All right. Happy Thursday. Welcome everyone to another session of the Virtual Environmental Ergonomics Symposium series. And once again, I'm Stephen Chung, and I'm co-organizing this along with Chris Tyler and Tadej Debevich. And today is going to be all about code and exercise. Ironically, now that it's kind of in Northern Hemisphere, it's starting to warm up. We're going to talk about cold. And it's going to be a fun talk today because once again, we're going to talk basic science and then we're going to really apply it to a situation, in this case, open water swimming. Just to let you know some of the upcoming talks that we have, we're going to be switching to in the coming months, a couple of talks on hypoxia physiology. So we have one on next week on June 11th, where we'll be talking about both nitric oxide availability and muscle metabolism. Then June 18th, we will be talking about pre-cooling in the heat. And this one is going to be the Asian Australian time zone friendly one. So that's why the asterisk is there. We'll have Rob Duffield from Australia, also Grant Landers and Mohammed Isan from Aspetar in Qatar, talking about different strategies for cooling prior to exercise in the heat. And just want to let you know, we had an unfortunate kind of cancellation for the June 25th session. So we are looking for individuals who are interested in speaking. So please, if you're on Twitter, direct message at ICEE 2021 if you are interested in speaking, or else email me directly at ICEE 2021 at gmail.com if you're interested in presenting the talk. Then we have, again, on July 2nd, we'll have hypoxia and metabolism. July 11th, we're going to talk about cross-adaptation, both the combination of multiple stressors such as hypoxia and heat acclimation. And then July 18th, we're going to be talking about the heat again. And the 16th, we're going to be talking about heat again, and specifically about some of the plans that was designed by the IOC for preparing for the heat in Tokyo 2020. Ideally, they'll still be in place for 2021 when the games are hopefully held. And again, we're going to be taking a break after the July 16th session so that we can all enjoy our summers. And we'll kick things off again in early September with a virtual conference that's going to be aimed towards graduate students and postdocs having an opportunity to present. So that's going to be the format for the next kind of month and a half for our sessions. Again, if you are interested in speaking on June 25th, please contact me. Again, direct message ICEE 2021 on Twitter, or else email me at ICEE2021 at gmail.com. So once again, today, we're going to be talking about cold. We're going to start off with a talk from Dr. Dominique Gagnon from Laurentian University, who knows a thing or two about living in the cold, based on doing his master's in Winnipeg in Canada. And where one of the famous songs is Portage and Main 50 Below, talking about one of the streets there in the wintertime. And he's also did his PhD in Finland, and now he's at Laurentian University in the northern part of Ontario. So he definitely has experience in the cold. He'll be talking about muscle oxidative state and metabolism during exercise in the cold. We'll then follow that up with Dr. Heather Massey, who's a senior lecturer at University of Portsmouth in the UK, talking about open water swimming and open ocean swimming, and both the science and the practicalities. Just want to note that Heather is also the host of a podcast called Humans in Extremes, where she's interviewed individuals including on ice swimming and on other extreme environments. And she promises she has some new episodes she's working on once courses are done for the summer. And there's, so you can find that at the website that is shown here, and also on most of your popular podcast apps too. So with that, I'm going to stop sharing my screen, and I'm going to let Dr. Gagnon take the stage, and the floor is yours.
Thank you very much for the introduction, Stephen. Really appreciate it. And again, really great pleasure to take part of this series of talks. We've had some great speakers so far. So from the introduction again, I'm located at Laurentian University. We have a beautiful campus surrounded by a lot of green, and it's a beautiful time of the year to be there. But a little bit about our community for those that never had the amazing chance to come and visit Sudbury. We are a very active mining community, one of the biggest in Canada. And surprisingly, we're talking about coal today, but there's a lot of research being conducted for heat stress in the mining in Sudbury. We have ultra deep mining projects with mines getting deeper every year, and so there's a lot of opportunities there. If you ever drive up, you should be able to see our big nickel by the side of the road. And we, as Stephen said, we have very interesting winters. Just 2 winters ago, we had 13 feet of snowfall, which if you are a winter enthusiast, it's a wonderful place to be. But if you do not enjoy the cold of the snow, this is absolutely horrendous. But as a whole, we do have a very active winter community. We have a lot of skiing going on, a lot of hiking, biking, fishing, hunting, skating. We have a lot of individuals being very active in the cold. Another example of that, every year we do have our hockey on the pond tournament on Ramsey Lake Road, which is one of the main lakes centrally located within the city. We have hundreds of hockey players coming from around the region, whether it's 0 degree outside or minus 30, people skate for 2, 3, sometimes 4 days in a row, and you stay pretty active. So just a brief outline of what we're going to talk to today. So we're going to first get through a quick overview about coal metabolism across different states, primarily resting and exercise. And of course, tissues, because metabolism does differ across tissues. And again, this is just going to be a brief overview before we get into human metabolism during exercise in the cold. And then we'll link that up to oxidative state in the cold, and then just finish it off with some recent advances that we have in the field. So first and foremost, as most of us know, has been exposed to cold within a few minutes of cold exposure, or if you have a slight drop in core temperature, very minimal, you do start to shiver, of course, and shivering contains both continuous and high intensity shivering. And again, both type of shivering relies on different muscle fibers. And so while continuous shivering relies more heavily on type 1 fibers that are more lipolytic in nature, high burst, high intensity shivering relies more on glycolytic fibers. And so how does that translate into actual metabolism? Since shivering is limited in its impact to produce energy, or if you put it on a scale of a VO2 max, for example, it doesn't really go beyond 20 to 25% of VO2 max. And so if you're looking here on the upper panel, shivering relies more heavily on glucose and glycogen. So lipids contribute very, very little. And on the right side of the red line here, as exercise intensity progresses, we have a completely different response in tissue metabolism, where lipids do increase progressively all the way up to approximately 50 to 60% of VO2 max before it starts declining, whereas carbohydrates keep increasing exponentially. So a very different response between resting in the cold and classic exercise substrate agitation curves. Beyond shivering thermogenesis, we also have non-shivering mechanisms. And in the past 10 to 20 years, we had tremendous advances in terms of broad adipose tissue activation with just mild cold exposure and the integration of UCP1s in BAT. And so uncoupling proteins are specific proteins, again, that are embedded within the inner mitochondria that is using the proton leak as opposed to producing energy from mechanical work. It's simply producing heat, and they are nicely activated during mild cold exposure. But beyond brown adipose tissue, which has been somewhat the main key in the cold for resting under cold stress, we now also know that within skeletal muscles, we have another isoform of uncoupling proteins, uncoupling proteins 3, which uses a similar mechanism. But if you look on the left-hand side here of the pictures, you have those large droplets within the cell. And you can see the location of the mitochondria kind of stuck all around the droplet. So really, really close by, because it does tend to rely more heavily on beta oxidation as opposed to glycolysis. And so different UCPs can use different types of energy depending on which tissue they're in. And Again, UCPs are have a decent variety of structures and across from the 1 to 4 that we know. So UCP1, as we said, found primarily in brown adipose tissue, has a high rate of glucose disposal, as we know. UCP2s, which can be found in other tissues, such as lung, livers, or even the intestine, for example, rely more heavily on glycolysis. UCP3s, which we just discussed, again, found not just in muscles, but cardiomyocytes and a little bit in brown adipose tissue. And UCP4, which we know a little bit less about, but these are found in neurons. So again, we have a wide variety of tissues that under mild cold stress have the ability to generate heat. And I do enjoy providing a good visual of what's going on at the mitochondrial level. And so this is a really fantastic figure from Brent et al. Just from 3 years ago, that from nuclear imaging, they were able to map out a mitochondria. And you can very clearly see the outer membrane, which is the filament surrounding the entire structure. And inside the lines that you see in there are the inner membrane. And we know that on the outer membrane, this is where you find most of your transport and binding proteins to help substrates make their way within the mitochondria, whereas the inner membrane instead, this is where you do find not only your mitochondrial complexes for oxidative phosphorylation, but also your UCPs. Now, why is that so important? We know that the reason it's so exciting when you're exposed to cold and why you want to increase that energy metabolism is that when you're looking at a young versus an older model of mitochondrial structure, when you're looking at the old, and in this case from the Brent group, I do believe they were cardiomyocytes from mice, both the outer and the inner membrane tend to change significantly. We know in older individuals or in a disease state, such as metabolic diseases, mitochondrial efficiency is severely decreased and oxidation of substrates is also compromised. And so both the outer membrane, which tends to lose its shape, either becomes more circular or becomes more lumpy, again, becomes less functional, and likely, again, the inner membrane, which tends to start overlapping over one another. So there is some significant changes that do occur at the mitochondrial level, whether, again, using an older aging model or a disease model. And as a consequence of that, using, again, a cold model, we have, again, a variety of groups, and this is just a few snips from just the past few years only, where we're really targeting fat, mostly brown adipose tissue with cold stress, to see how we can really reactivate those mitochondria the best way we can, right? How can we use cold, even just mild cold exposure repeatedly, to hopefully activate this browner tissue or even browning some fat in order to increase our energy expenditure, mitochondrial health, and maybe try to deal with mitochondrial dysfunctions and even obesity. But I think this has been really well laid out by Carpentier recently, where if we just look at glucose disposal, so just glucose by itself, and if we take very active brown adipose tissue into an individual during mild cold exposure for 24 hours, the quantity or the absolute volume of glucose disposal is approximately 3 grams, which is not that much, as opposed to the glucose disposal that's being performed in skeletal muscles. And so just based on sheer volume, despite the fact that we do want as much tissue to be metabolically active, skeletal muscles, just because of volume, have a much broader impact on human metabolism. And so this is one of the reasons why in our lab we try to combine both cold and exercise. How can we really get the benefit of it all? From resting metabolism, we do know that that either feeding or shivering or non-shivering thermogenesis can only marginally increase energy expenditure, whereas when you are exercising, again, you can crank it up to all the way to 1,000 watts or so. On the right-hand side, you may have noticed the Montreal Naked Man, which I think is a wonderful example of an individual exposed to cold. And so a few years ago, he made the news running naked in the northern end of Montreal in the middle of the night, where it was approximately -15, -20 degrees Celsius. And this just shows how powerful the human being is to be able to produce and generate a high level of heat in these conditions. And this is also representative of, I think, what a lot of labs conducting cold research experience, where it is very challenging to ask individuals to remain still under cooling protocols. And this was discussed previously by one of the speakers, where behaviorally, to maintain thermal regulation, as soon as we get a little bit cold and we feel cold, we want to move. And so how can we better integrate more tissue, more mass to be metabolically active and to remain active? So again, by combining both exercise and the cold, this is what we've been examining for quite a while now. So this is one of our first piece of work from many years ago. So what we did in this case, we did look at exercising or brisk walk at 50% of VO2 max, low intensity, or running at 70% of VO2 max in both a cold environment that's set at 0 degrees Celsius or in thermoneutral conditions set at 22. And one of the main purpose of this work was to match VO2, because one of the issues that we faced in the past was that a lot of protocols were based on matching workloads. And we know workloads under different thermal stress can lead to different VO2, and we know that VO2 is a core regulator of human metabolism. And so by matching VO2, we did notice an increase in fat oxidation within each exercise intensities between cold and thermoneutral, and those translated in approximately 1 to 2 kilocalories per minute. Now, just to be clear on the results here, this is not an additional energy expenditure. This is simply transferred from carbohydrates to fat. But again, having a greater fat turnover has been linked to many metabolic benefits, and this is what we've been gunning for. Another piece of work linked to what was just presented, we were also interested to know whether or not shivering preceding a bout of exercise would potentially influence fat oxidation. The reason is, we've just discussed earlier that the metabolism of shivering differs significantly from the one of exercise, where glycolysis is the main driver for energy production. And so this study, what we've noticed is that with pre-exercise shivering, there was greater availability of free fatty acid, more catecholamines in circulation, but despite these changes, metabolism remained the same within, again, either low or moderate intensity conditions between shivering and non-shivering. And so which led to the idea that this might be a more intramuscular process as opposed to a systemic one if we're changing substrate availability but no changes in oxidation. From there, we started to look instead at not just specific exercise intensities, but at this point across the entire spectrum of exercise intensities, and look at the entire oxidation curves for both fat and carbohydrate. Again, as I recall, we know the curvilinear nature of fat, where, again, in a more parabolic shape, starting from very low rates at the beginning of exercise, and then increasing until midway through the exercise intensity, and then dropping again towards the end, whereas carbohydrates instead is more exponential in nature and just increasing through exercise intensity. And what we've noticed as we're conducting our exercise under cold and warm temperature, in this case, again, cold was still at 0 degrees Celsius and warm was at 35 degrees Celsius, is that high rates of fat oxidations were maintained even in later stages of exercise under cold stress, which was definitely not seen in the warm. And if we actually look more closely at the curves themselves, we do see that in the cold, the curvilinear nature of our fat oxidation curves remains, but in the warm environment, it also tends to disappear and becomes more linear. So just the nature of the curve disappears. And so we ran a mixed effect regression to see if temperature has an effect on shifting metabolic curves, and the answer was yes, it did. Another metric that we also enjoy using in our lab that's a little bit simpler sometimes to understand certain concepts is maximal fat oxidation. We know that MFO has been associated with insulin sensitivity, training status, blood pressure, metabolic profile, and other cellular markers of health and metabolism. And so the way to do this, once you have your curve, and whether it's mathematically shaped or you're using raw data, what you do now is simply pick the peak of your curve, and it gives you your peak rate of oxidation for fat during a specific exercise bout. So from this technique, what we've done is examine during treadmill and cycling in both men and women changes they could have in their maximal fat oxidation. And as you can see from the graph here, unsurprisingly, we did notice increases in maximal fat oxidations in essentially in all modalities and between conditions, again, between warm and cold. And similar to previous work, We did notice, again, the increase summed up, if we transfer in grams per minute of oxidation for fat, it does end up to 1 to 2 kilocalories of fat per minute of a change, which mimics what we've seen in the past. And again, so if you go to exercise for a half hour, an hour, you spend 2 hours outside being physically active, you can look at a significant shift in energy expenditure towards fat oxidation. Most of the work in the past that looked at an increase in fat oxidation and the cold has been generally linked to beta adrenergic response and just overall lipolytic response. And so this is something that we've known for a long time. We are aware that an increase in circulating catecholamines will activate beta adrenergic response, and then through cellular signaling pathways, starting from cyclic AMP all the way to hormone-sensitive lipase, it will start breaking down triglycerides from lipid droplets in order to release glycerol and fatty acids to then circulate systemically to be used as substrates. And so this in itself has been pretty clear from a long time. The one issue that we're facing now is that exercise by itself also increases catecholamines significantly. And then if we add cold by itself increases catecholamines, but then and beta adrenergic response. But then again, if it's matched with exercise, the increase sometimes is exactly the same. It doesn't change. And the same, if we start to look at heat, the same is also, the same is seen. So heat will also increase catecholamines. And so overall, if you just add any kind of stressors to the mix, it just kind of mixes itself, and catecholamines can rise to pretty much the same extent. Doesn't matter how you combine or remove some different types of stressors. And so it gets really mucky in that way. So we can't really fully rely on just simply saying that it's beta energetic response that drives the extra lipolysis. It could be the case, but there might also be other modulators. And so one thing that we've done in our lab is also look at muscle oxidative state. So what's happening from an oxygen standpoint in your muscles in the cold, and how potentially could this be a driver or a change in metabolism subsequently. And going back to previous and slightly older literature, we all know that as you are increasing exercise intensity, whether you're looking at a workload or absolute VO2 or relative VO2, and whether you're looking at systemic or local blood flow, you should have more or less a linear increase between load and flow. Also, in thermoneutral conditions, we also, as if you're looking at tissue oxygenation within the first few minutes of exercise or in the very first stages of increase in exercise, there's a slight increase in tissue oxygenation level, but then it starts to drop progressively as exercise intensity increases. And this is a result from, despite an increase in blood flow, a greater uptake in oxygen in the cell. And from the little bit that we know in the cold, if we're looking in the middle panel here, we can see that the oxygenation level from a drop in tissue temperature tends to be seen, again, it tends to be deoxygenated at the very early onset of exercise, but then at steady state exercise is fairly maintained. It doesn't change that much. And on the right panel, if you're looking at total hemoglobin or a measure of total volume of blood, it's not quite a measure of blood flow, but it gives you an idea of the total volume of blood that's actually within the site of that you're looking into. In this case, you're also looking at a drop, but this drop is maintained. So you do have a lower blood volume within a specific set of muscle tissue during exercise. Other factors that we know that do regulate oxygen content within the muscle or the movement of oxygen. So we know that diffusion coefficient of O2 is decreasing at approximately 3% per degree Celsius. That drops primarily from a shift in the intracellular milieu viscosity and the kinetic of energies. And despite an increase in gas solubilities, for oxygen, it is 1.4% per degree Celsius, you still have a drop in the diffusion constant from the capillaries to the actual muscle cells, or about 1.6% per degree Celsius. And we're also, of course, well aware of the oxyhemoglobin saturation curve, where oxygen binding becomes an issue with cooling, where the unloading phase of oxygen to the cell becomes more challenging as the tissue is getting cooler. And so overall here, what we just want to show is that there are new challenges with cooling of just, regardless of the amount of oxygen that's actually on the site to be ready to be transferred to the cell, just the transfer itself is already compromised. And so if we gather all this together, we start to figure out that not only there's vasoconstriction in the muscle, which automatically provides lower O2 supply, lower O2 diffusion coefficient. We know that there's a higher VO2 demand at a fixed workload when the muscles are really cool. Overall, lower tissue oxygenation level, and all these from previous work, we know that it should lead to an increase in carbohydrates. So this is what we were essentially that we should expect. So this is what we looked into. But at first, we wanted to get more insights about the actual oxidative state, and so we had a few studies to look at see how those changes do occur under various conditions. And one of our first study was to examine the effects of 3 minutes leg or knee extension on a dynamometer at 10, 30, and 50% of maximal NVC, and then looking at changes in blood flow and VO2. And this was done using near infrared spectrometry as technology to assess that with a cuffing system, as well as using intramuscular probe located within the vastus lateralis to get a measure of muscle temperature before, during, and after exercise. Our cuffing protocol here that we use, this is our way that we assess blood flow and VO2 non-invasively. To get a measure of blood flow, you need to look at your at a circuit in a way that you want to make sure that blood can enter the leg, but nothing can come out. And in that way, with a pressure of approximately 80 millimeters of mercury, which is the pressure that a cuff applies proximally to the nearest unit, in doing so, again, you do allow arterial blood to come in the leg, but you do block venous return. And so if you look at the signaling from the nears right below on the left-hand side, so the green line or total hemoglobin again is, in this case, representative of flow to a certain extent. And if you take a chunk of the slope, you can subsequently calculate the blood flow for that particular area. On the right-hand side, if you're looking at VO2, this time your circuit's got to be closed. Sometimes there's nothing coming in and nothing coming out. And as a consequence of that, if you look at the green line again, you can see that total hemoglobin is fairly straight. And if there's some slight fluctuations, whether or not it's from a shift in pressure, this can be corrected afterwards. But to do so, you need a much higher pressure that is far superior to the arterial pressure. And so the cuff is generally set at approximately 300 millimeters of mercury to make sure that nothing comes in, nothing comes out. And then you can use either the shift in oxygenated or deoxygenated hemoglobin content. And the key feature here is that within an environment where the total volume of blood is not changing, the only thing that's going to change is how much the nearby cells will use oxygen for energy production. And so for O2HB, if you take a chunk of that slope again and use that, and you can mathematically calculate VO2. And so from that work, what came out is that the original relationship that we have with workload or VO2 or MVC in our case with blood flow or QM on the left is that heat disproportionately increased flow and only did so marginally under cold stress. And again, in this case, we had our muscles being heated by 3 degrees Celsius and our muscles cooled by 3 degrees Celsius. And when we looked at the VO2 results afterwards from that shift in blood flow, even though the blood flow was far superior in the heat, it only resulted in a small change in VO2, but it was different between heat and cold. And so both the relationship of VO2 and flow is slightly altered across exercise intensity. Then we took that work and moved forward in looking at oxygen kinetics. And the reason we want to look at the kinetics is because it does provide a good window of metabolic adjustments from rest to steady state. And again, when you look at kinetics, you want to look at the curve from the start of exercise until you reach a steady state. And within that curve, you can look at a few features. And so you can look at the time delay, whether or not it takes a long time for the body to respond from the start of exercise to start adjusting to the metabolic demand. You may also want to look at time constant, or T, which is reaching 63% of the entire response. So it does provide a significant amount of windows. And again, it is a useful tool to assess some limitations or possible limitations of energy systems under cold stress. So for that study, we use a thermoneutral condition, of course, and then we had one condition where we cooled the muscle, which again, we wanted to have those biochemical changes locally only, as well as some local vasoconstriction, and see how those shift in local oxygen delivery and diffusion could impact our kinetics. And we further wanted to see if core cooling, which we're expecting to have a change in cardiac output, so more systemic oxygen transport, as well as potentially shivering and a shift in pulmonary VO2 could further influence those kinetics. So before we get to those curves, just a couple of key points to guide the discussion here. So we did find an increase in VO2 from whole body cooling. We did see a decrease in cardiac output from whole body cooling, and both whole body cooling condition and muscle cooling condition led to decrease in local blood flow. But how did that translate into our actual kinetics or our energy system to respond? And the answer is that it did not. So we're quite surprised by this. We're either looking at time delay or our time constant of the curve, and it didn't matter which condition that we were, all three curves were different. You can probably visually assess the fact that the curves for the whole body cooling condition tend to be more elevated simply because VO2 was already elevated from shivering, but the shape of the curve remained the same. And in some ways, I mean, the message that we can extract from this is that despite lower blood flow and despite limited diffusion capacity, within the work rate that we've employed, the energy system is not compromised to a point that you can still successfully perform physical work. So from that study, what we did, we also moved on to look at specific muscle temperature and how did that translate subsequently to actual metabolism. This is a study that actually just got published today. Timing was phenomenal on that one. And we employed similar techniques with NIRS and intramuscular temperature probe to make sure we had the right muscle temperature. And what we did, we employed a dual leg cooling protocol, and we compared that to a match non-leg cooling. And immediately after the muscles were cooled by 6 degrees Celsius, we initiated exercise at 70% of the ventilatory threshold. And this was maintained until they reached 3 degrees and 3 degrees below baseline, up to baseline. And then this was followed for another 30 minutes afterward. And so we had measures of different temperatures as well as continuous exercise afterwards. And what we found in this case is if you look at the muscle temperature in the middle of the screen there, so it evidently increased with exercise, which is something we expected. The increase was a little bit more severe following cooling, trying to return more quickly to baseline values, but it didn't really return to baseline values for a long time. So it was different from the beginning. At 3 degrees, very different again at 0 degree. And again, it took a long time before it got back to baseline until approximately the 15-minute mark to go back to the same temperature. But in this time, what we saw is a clear shift in tissue oxygenation. It was significantly lower in the muscle cooling protocol. pH was lower, partial pressure of O2 was lower, partial pressure of CO2 was higher. And essentially what we had is, despite the fact that these are all drivers that should have increased carbohydrate oxidation, we found no changes whatsoever in any of our substrates. And this indirectly somewhat suggested a muscle temperature dependent mechanism that still needs to be explored. For the last section of my talk here, just a couple of side pieces that we have that kind of complements what we do from various students. We also took the opportunity recently to look at exercise in the cold under HIIT type exercise. And so we used, if I recall the specifics, it was 8 to 10 repetitions of 1 minute at approximately 90% of max VO2. In this case, with match VO2 and energy expenditure, we also did see under that type of exercise greater oxidation of lipids and lower oxidation of carbohydrates, which we're very pleased to see, because we're sure at the beginning, since the demand of the metabolic demand at these types of intensity primarily rely, if not solely almost, on carbohydrates, but there was still greater fat oxidation regardless. When we examined other markers of metabolism afterwards for lipid profile, insulin, and glucose, we didn't find any differences, but instead we did take the opportunity to look at post-prandial metabolism the next day. And so following that acute study in HIIT, because we know that HIIT exercise compared to moderate exercise has a more beneficial outcome on post-prandial metabolism, which again is the metabolism following a meal. We wanted to see if that extra shift in lipid metabolism from the exercise itself could maybe translate to a greater shift in fat metabolism as well the next day. There's also a great meta-analysis that just came out in APNM from Pearson that at the same time has demonstrated a nice moderate effect of acute exercise on postprandial metabolism that again, that's nicely regulated by exercise. And so we took that opportunity to look at our results the next day for a 4-hour postprandial time. And what we've noticed is no differences in oxidation rates whatsoever. But surprisingly, what we saw was an increase in glucose during the 4-hour period. And so that increase over time somewhat suggested that the glucose-sparing mechanism that we may see during the exercise bout in the cold the day before might actually have an influence on glucose regulation the next day. Either it tries to complement by increasing carbohydrate oxidation the next day, or another mechanism might be at play. We're not sure, but there is something occurring postprandially following exercise in a cold environment. We also looked very briefly at how potentially exercise in the cold could influence metabolic flexibility. Metabolic flexibility is the ability of an organism to switch back and forth between carbs and lipids depending what it needs. And so depending how much exercise you do, if you maintain an energy balance, if you're exposed to a variety of environmental stressors, levels of hormones, tons of regulators really modulate how mitochondria are flipping between lipids and carbs. And now that we have this increase of this shift in fat oxidation in the cold, one of the questions that we had was whether or not this potential shift could, in a prolonged way, maybe shift to a glucose toxicity or other kind of toxicity limiting mitochondria's ability to switch back and forth. And so what we did, we used a glucose loading versus non-loading protocol and assessed that difference in both a cold and thermoneutral environment. And the metrics that we used for this one was a shift in MFO, fat max, the crossover in energy expenditure between carbs and lipids, as well as a difference in respiratory exchange ratio. And we're pretty pleased in this case that not only the cold increased fat, but the glucose loading also increased glucose levels the same way in both cold and thermoneutral. That led to, again, no change between conditions, potentially suggesting that the cold and metabolic pathways related to the increase that we saw in glucose oxidation could be mechanistically independent, similar to the other studies that we saw. And just to finish off, if I can leave you on just a couple of words, cold exposure does impact the oxidative state of skeletal muscles, right? It does lower oxygen supply, likely lowers oxygen uptake as well. But nonetheless, there is this increase in fat oxidation that we keep seeing over and over again that could be very meaningful and provide benefits metabolically. And along with the many students and staff that came across the lab over the past few years, I want to thank many of my colleagues, Stephanie Munton, a wonderful research assistant, my grad students, and my funding bodies. Thank you.
That was great, Dom. Thank you very much for that talk. And some of the questions I've been seeing, one of them is, I guess, more of a contextual question. One, like the work you've done has looked at actual whole body cooling or actual muscle cooling. So, however, like you have in that runner in this slide here, he's dressed appropriately, and he's probably not really having a significant whole body kind of cooling. So, can you kind of talk about that dichotomy? Obviously, with what Heather's going to be talking about, where you're in water and you have minimal clothing, you are going to cool down quite significantly, or have the potential to cool down significantly. Whereas, most of the time, you're going to be dressed appropriately, where you're not going to have significant whole body cooling.
So that we're clear, when you talk about body cooling, most of the protocols that we employed, and you're absolutely right, we normally use people that are lightly clothed, just shorts and t-shirts in 0 degrees Celsius. And for pretty much all the studies within that temperature range, even at light intensity, we see either a perfect maintenance or even an increase in core temperature. So there's not really a drop in core temperature per se. The only thing that does cool down is peripheral tissue. And one of the protocol that we use with leg cooling, we really want to have deep cooling of muscles themselves, but not necessarily a shift in core temperature. In case of the classic runner that we see here again, so there are evidently areas of your body that will remain cool. So of course, most people, even when they're well dressed, hands, feet, and of course your face is uncovered. And for most people that are running in the wintertime, I think can agree, after running 5 minutes, you're already starting to get warm. And so you're starting unzipping and your neck is open. And so you have this change in clothing as you're exercising. Now, since we're not aware of the exact set of pathways that do regulate these responses that we see right now, I can't really comment on that. But if it's partly based on beta-adrenergic response, then just activating the trigeminal nerve in your face, again, which can regulate catecholamines release or beta-adrenergic responses, is already a significant stimulator. So that is one. There's been studies where individuals were fully clothed with just their face wide open and facing cold wind, and there's significant changes on cardiovascular responses, respiratory responses. And so if we make the assumption that these responses are affected, then there's likely something influencing metabolism as well.
All right, great. And we have another question from Chris Minson talking about, the studies that you've done, and the large majority of studies about exercise in the cold have been acute studies, interventions. Can you comment on what is known about, whether there is kind of cold adaptation, or is there potential for this, or what that process is?
So the current situation right now is preventing me to answer, simply because the study we're supposed to run this spring has been postponed, but we have currently limited understanding in that regards. There's been a few studies that have looked at post-exercise limb cooling, for example, following, I think it was an 8 or 12 week training protocol. And so we have cooling post-exercise, not necessarily whole body cooling, but limb cooling post-exercise. But I do believe whole body cooling post-exercise repeatedly, or cooling during exercise, and the adaptation through that. Currently, I'm not aware of anything that has been published that I don't think there's anything in the moment, and it's something we're currently working on, because we are expecting to see, if we have this shift in metabolism that is so acute and repeated every single time, there should be something chronically as well. Whether or not that translates into functional changes that we would like to see, then is something that needs to be answered.
Okay, and I think I may have missed it, but I also got a message with another question about the kind of, I know you've taken some muscle biopsies. I can't recall whether you really showed that data or not, and then looking at both kind of, I guess, muscle fiber recruitment, and also kind of some of the glycogen content of the muscles.
Yeah, so there has been, and again, so I'm definitely not the first to demonstrate the increase in that shift in metabolism in the cool, all the way back from 75 from Fink work. We already knew from biopsies, but not consistently anyway, that there tended to be a greater use of intramuscular triglycerides, and some glycogen was saved. There's other groups that have demonstrated some similar responses with using biopsies. But the issue that we had, again, a lot of time, it was either the reason why there was no differences in some other studies is, again, VO2 was not matched, or the cooling protocol was inadequate. And so we have a lot of different models that we had to play with before we got to the one we're in right now. And again, because we're a young lab, so we just started to take biopsies this year. And so this is definitely the next stage of what to look into. And just to lick on that, I know we have speakers coming in the next few weeks. I think that should be speaking about limb cooling and mitochondrial changes as well. And so I think this is better left off for them, but there's a lot of work to be done in that field still.
All right, great. So again, thank you very much, Dominique. That was an excellent talk, and I think you'll be a great resource also for future viewing too. We're going to ask you to stop sharing your screen. And with that, I will ask Heather to share her screen. And Dom, there's a couple of outstanding kind of Q&As. Maybe you can take them while Heather is speaking. All right, Heather, the floor is yours.
Can you see my screen there, Stephen? I'll take that.
Yes, we can.
Yes. Okay, thanks very much, Stephen. And thank you very much, ladies and gentlemen, for attending this talk. And thank you, Dom, for an excellent talk too. This talk really is about highlighting a challenge I undertook last year, which was to swim the English Channel. So swimming from England to France. And to do so requires just the wearing of a single swimming costume, goggles, and also swimming hat, and your choice of grease. Now, the swimmer, as pictured, as shown in this picture, stands on the beach in Dover in the south of England and swims to the pilot boats, which you can see pictured here. And hopefully they get the opportunity to see the French coast just before they leave Dover. Now, this talk really is about an English Channel swim, but it equally applies to any long distance open water swim, normally in sea conditions. And it's really about what it takes to get to the start line. So we're going to talk about some of the science and some of the practical safety aspects of cold water swimming. Now, I had always imagined if I was ever going to swim the Channel that this was the picture that I was going to have, in my view, when I stepped onto the beach in Dover, waiting to start that swim. It's a little bit far from reality, in actual fact. It looked a little bit more like this. Now, this is actually Sarah Thomas starting her swim last year. Unfortunately, it was even blacker when I started mine. So you swim from the pilot boat onto the beach in Dover, and you stumble ashore because you can't see anything, and then sort of belly flop back into an inky black sea, which again, you can't see any of the waves breaking over your face. And so for the first couple of hours of my swim, it was really pitch black. And it's really quite an scary experience. It's very dangerous and very disorientating, and not something that should really be attempted without a very experienced pilot and crew, and with some prior training as well. Now, a lot of long distance swims do actually start in the dark because the wind speeds tend to be lower, and so the chop of any sea tends to be less. And you'll notice on this slide that it actually says this was my second attempt to start the swim. Now, the first attempt, unfortunately, we didn't even make it to the beach. We got onto the pilot boat and were exiting Dover Harbour to come onto the beach, and I was already stripped and ready to go in preparation for swimming onto the beach when the pilot said, "We can't swim, it's too rough." So the health and safety aspects of this sport are really very important and are taken very seriously. And as a swimmer, you put your body in the hands of experts. And I'll come on to talk about the pilots a bit later on. But one pilot describes long distance open water swimming as an extreme endurance sport ruled by the elements. And that's exactly the fact. When you swim, it's entirely up to the pilot who indicates when the weather and the wave conditions will allow you to make that attempt to swim. And we don't have a fixed date for swimming the Channel. You have what's called a weather window, and so you're on standby for the entire period of time of that weather window. You have really no control. Once you get to the beach in Dover and you want to start your swim, you hand over control of everything else to your pilot and to that crew. The only thing the swimmer has that's able to control is actually putting one arm in front of the other and swimming. So swimming the English Channel is really a physiological and psychological roller coaster. And for those of you that have not seen an English Channel swim before, what the swim involves is swimming from a town on the south coast of the UK called Dover, and you swim from one of the beaches just outside the harbour, swimming ideally to and landing on the Cap Gris-Nez in northern France. Now, the swim is never going to be a straight across, and so the shortest possible distance is 21 miles, but your swim might be slightly longer than that. The first person to attempt this was Captain Matthew Webb, and he completed it in 21 hours, nearly 22 hours. And so far, there have been nearly 2,000 successful solo attempts. And now the numbers vary, the success rate varies between seasons based on weather conditions, and there's lots of different reasons why the success rate varies over the seasons. But I was really fortunate and very lucky to be able to land the swim right on the Cap Gris-Nez in northern France. It took me slightly longer than I was hoping at 16 hours and a minute. And again, I didn't get the opportunity to land on a beach, which I'd always imagined I would. I ended up landing at the base of a cliff and climbing onto a rock to wave at the pilot boat. So this is the final photo the crew were able to take because they couldn't get any closer into the water, into the cliff to take any further photos. So this talk will primarily discuss how we get to the point where we can stand on the beach in Dover. And I liken the talk that I'm going to give to a little bit of an iceberg. Although there's no icebergs in the English Channel, it's not that cold. The idea that an iceberg is, there's some of the iceberg that you can see, and they're the factors that you know about. And then quite a lot of the iceberg that you don't see are those factors that either you don't know about or can't control. And that is very much like swimming a long distance swim. And so one of the first factors that people are more aware of is that the water is cold. Now, Dom gave us a very good introduction to cold air, but cold water is slightly different. We know that cold air is actually quite a good, well, air is quite a good insulator. We can rapidly heat a boundary layer of air and warm that air to reduce any transcutaneous heat loss. And we know that water is has about 24 times the thermal conductivity of air. And the energy required to heat a given volume of water is about 3,400 times that of air. So that is the product of density and also specific heat. So when we come to look at the cooling power of water in terms of deep body temperature, it has about 3 to 4 times that of the cooling power of that of air at the same temperature. And for a given heat loss, it can be achieved at a higher temperature in water and for a narrower skin to environment temperature gradient in water compared to air. So what was the water temperature in the English Channel? So all these water temperature readings are taken from a buoy called the Sandetti Lightship Buoy, which is right in the middle of the English Channel. And we'll see here from the graph, we've got our water temperature on the vertical axis and time on the horizontal. We have the blue area on this graph is the normal swimming season for English Channel swims. So you'll see that my first attempt was just outside of the start of that swim zone, and the second attempt right at the very start. So the water is still warming up, and the vast majority of my training in open water was conducted at temperatures considerably cooler than that. So now we know that the water is cold. So what effect does that have on the body? Well, initially when we go into cold water, we experience the cold shock response. And that's really a rapid skin cooling, which is sensed by the cold receptors right in the very surface of the skin. And that causes a sympathetic stimulation, causing an initial gasp and hyperventilation, as well as vasoconstriction, tachycardia, and increased cardiac contractility and increase in blood pressure. Now, for those people that are not accustomed to going into cold water and may accidentally be immersed, and they're not able to control their breathing in time with having their airway above the surface of the water, this may lead to aspiration of water and potentially drowning. And also, for those that have underlying cardiac conditions, if we have a rapid increase in heart rate and also blood pressure, this adds to the work of the heart and can result in adverse cardiac events. So this is not something to be taken lightly. It's an extremely dangerous response. But we know that it can peak within about the first minute, and that the response decays thereafter. Now, a far more insightful diagram about the mechanisms of cold shock response can be found from Tipton's work in the late 1980s. But what we do know is that the cold shock response is not a thermoregulatory response, and that we can habituate to this response. Now, we can habituate to this response by repeatedly being immersed in cold water. And again, research in the 80s found that actually the ventilatory response and also the heart rate response was considerably reduced by repeated immersions in 15-degree water. We also know that the habituation to cold shock has quite a long period of decay, and that we can have a look at two groups here. We have a control group and also a habituation group. And that control group were not immersed for any other period of time other than those immersions you see on the graph. Now, the habituation group were put in cold water repeatedly between day 5 and day 1 and day 5 on this graph. And we see that the inspiratory volume here of our volunteers was the same on day 1 for both the control and habituation groups. But by day 5, we see that the habituation group had reduced that inspiratory volume response. And that response remained right up to 7 months post that initial habituation. So that response is quite a long-lasting response. So again, we know we can habituate to that cold shock response. But another question that quite frequently people ask is about the psychology of going into cold water. And there is a psychological component potentially to going into cold water. Now, what I've done here is I've combined several studies from our laboratory that have looked at the potential for a psychological component. So what we have here is the inspiratory volume during the first 3 minutes of immersion in cold water at 15 degrees Celsius. And there are a number of different groups put on this graph. The first group is actually, the first two groups here, the blue and the orange group, are actually the same group, with the blue group being the first immersion and the orange being the 6th immersion of that same group once they've been habituated. And we see there that inspiratory volume is reduced quite dramatically within that initial period of cold water immersion. We also see that this grey group here, this third group here, this is a group that have been habituated to cold and then have an anxiety induction on that final 6th immersion. And we see that anxiety induction has very little impact on their inspiratory volume during that final immersion. However, if we repeatedly immerse people and maintain that anxiety induction in all of those repeated immersions, we see that there is some of that cold shock response that actually remains even though the volunteers have been repeatedly immersed in that water temperature. So there may possibly be a psychological component. We also know that from breath hold studies that have used psychological skills training, that we can actually increase the breath hold time during a cold water immersion just by using psychological skills training. So it does appear that there may be a psychological as well as physiological component to the cold shock response. So the next component that people know about is that actually swimming from England to France is a long way, or any other long distance swim. It's a long way in cold water. And yes, it's right, yes. We have in this photo, we have Trent Grimsby, who has swum the English Channel in the fastest time at 6 hours 55 minutes. Jackie Cobell, who's had the longest swim, single swim in the Channel at 28 hours 44 minutes. And then last year, Sarah Thomas completed a 4-way non-stop crossing of the English Channel in 54 hours. So swimming the English Channel takes a long time and you're in reasonably cold water. But when we look at some of the survival curves that are potentially used for search and rescue, we see that the survival time in water temperatures that are fairly similar to those that cold water swimmers experience. Now for the English Channel, I've identified these by these red triangles. That's between 14 degrees Celsius and 18 degrees Celsius. Now some of these survival time curves really differ in their prediction about how long people can survive. Now some of them will depend on whether people are wearing life jackets, different types of clothing, but the predictions are anywhere between 1 to 2 hours, right up to 10 to 16 hours. So there are some differences of opinion about how long people can survive in cold water. However, cold water swimmers not only survive, but they thrive in those conditions. Although cold swimmers tend to choose the best conditions in order to go and swim the Channel and not the worst, there are still some differences between cold swimmers and a normal population. So let's just have a look at those in a little bit more detail. Again, some research from our lab looked at the change in rectal temperature during cold water swimming. So just for comparison's sake, I've included a 90-minute swim undertaken in 25-degree water, which is just a little bit cooler than normal pool temperature. And we see there's quite a variety in the deep body temperature response of our 10 volunteers, from those people that were a degree warmer at the end of that 90 minutes to those that were almost a degree colder at the end of the 90 minutes. But what we also see is that they've all appeared to have reached thermal steady state. So the the heat loss that they have from the water appears to be also equivalent to the heat gain the metabolic heat production that they have from their swim. So let's take the water temperature down a little bit. So 18-degree water temperature, very similar to that experienced by swimmers in the English Channel. And the picture is quite different. Again, we have the change in rectal temperature of our volunteers. And again, we have a cohort of people that appear to be in thermal steady state for the duration of their 90-minute swim and actually have higher deep body temperatures than their baseline levels. So they appear to be in thermal steady state. Their metabolic heat production is matching their heat loss. However, there were a number of people that either cooled significantly or withdrawn because their heat loss was much greater than their metabolic heat production. And so cold water swimmers really need to be able to retain the heat within their body in order to carry on swimming for prolonged periods of time. And so they really need to be within this top group here, where they're able to maintain thermal steady state for long periods of time. So how do we do that? Well, in order to maintain that thermal steady state, we need to be able to offset that water temperature, the heat loss from the water temperature, be able to offset by metabolic heat production. Now that could be through a fast swimming speed, so we're maintaining that high heat production, but it may also be having a level of fitness that we're able to maintain that high swimming speed for prolonged periods of time. In addition to that, we need to feed regularly to maintain blood glucose levels for muscular contraction, so we can maintain that high heat production. If we don't feed well, then glucose levels will fall and the swim speeds are likely to drop off and will fatigue. And that's the point where the heat loss may exceed the heat production. We may also need to increase our insulation as well, be that through the wearing of a wetsuit, external insulation, or internal insulation. So looking at some further evidence from research that we've undertaken in our laboratories, looking at some elite open water athletes who were swimming in 16 to 18 degree water for approximately 2 hours, looking at the rectal temperature or changing rectal temperature on the vertical axis. And what we really noticed was that the significant factors in this group of people were the skin fold thickness, their metabolic heat production against their body surface area. So we found that those that had very small skin folds, very low skin folds, had the greatest rate of body cooling, of rectal temperature cooling during those swims, whereas those with the highest skin fold thicknesses had the much greater, had the least change in rectal temperature. So whilst we may be able to increase our metabolic heat production by increasing our speed, if we're undertaking very long duration swims, it seems unlikely we'll be able to maintain that for prolonged periods of time. So I was left with the choice of trying to increase that insulative layer around my body. And looking at the right-hand side of this graph, I think you'll see that I managed to achieve that relatively successfully with within 6 months. I put on quite a substantial amount of body mass, predominantly body fat. And we'll see in a minute how that's affecting my deep body temperature during the swim. So now we're starting to look at some of the elements that are less well known about cold water swimming, or particularly swimming for long distances. And that's the feeding regimes of swimmers. Now, we have to feed during the swim in order to maintain that swim speed and prevent fatigue. But nobody on the boat can touch you, and you can't get off can't get onto the boat in order to feed. And also, you may start to consume a substantial amount of seawater. So the nutrition you get from your swim team is not the only source of fluid intake. So these things need to be accounted for when we're looking at feeding regimes. So in terms of feeding and training, what we have is quite a strict regime in terms of our training and combination of feeding. So I trained Monday to Friday, weekdays, twice a day for up to 3 hours in the morning, and then again up to 3 hours in the evening. And then weekends, long swims, which may be up to 8 hours in total. Now, during that period of time, I was consuming quite a large number of calories to offset not only that large amount of training, but try to increase body mass as well. So having 4 large meals a day, which did mean getting up quite early in the morning to make sure that I'd properly digested food before a heavy training session. In addition to that, we're looking at high calorie snacks, and while swimming, trying to adopt the feeding regime that I had during the swim. This diet was very high in carbohydrates, particularly high glycemic index carbohydrates, so an incredibly obesogenic diet, as we proved from the DEXA scans. Now, when we talk about feeding in open water swimming, it's useful to have a look at literature within the area, but there is relatively little in terms of the scientific evidence for type feeding strategies within that literature. We have a review paper from Shaw in 2014, and it basically suggests for very long swims that we have these are the potential problems: thermoregulatory problems, glycogen depletion, limited feeding opportunities, and also GI distress, which is going to happen if you're consuming a large amount of seawater as well as your nutrition as well. Ooh, sorry about that. And what we see also is some recommendations about things that we can do in order to feed adequately for the swim, but it's not very prescriptive. And so more recently, Kumstadt has documented a case study of one elite open water swimmer who took part in the FINA open water swim series during a winning season, and documented in quite a lot of detail the feeding strategy during the swims. Now, her swims were a little bit shorter in length, if not in distance, but she swam, take this first race here, where she swam for 8 hours, just over 8 hours. If I were to consume the same volume of fluid that she undertook, I'd be consuming around just over 11 litres of fluid in 16 hours, in addition to any seawater that I consumed, which seems likely would have caused me a great deal of GI distress. So in the absence of any science to support a feeding regime, you fall back on the opinion of experts, and there are many experts, and they all have very different ideas about the feeding strategy which is optimal. And so you have to find out what works well for you, and this is why you need to incorporate it into your training regime as a consequence, so that you are happy with that feeding regime and it suits your purposes. But one of the things, there are several things that the experts all agreed on, and that was having a meal well in advance of the swim that was properly digested and you're ready to go for your swim or your training session. In addition, that they suggested that feeds should be short and fast. Now, the reason for that is, particularly for an English Channel swim attempt, we know that every time a swimmer stops, that means that they are moving sideways and they're moving away from their goal, and that could add hours. It could add quite a lot of time to their journey if they are successful. And so we need to make sure that the feeds are very short to prevent the boat going sideways, and therefore they need to have small amounts of feed to make that a very fast feed. And I settled on a feeding regime which was around having about 150 mils of a carbohydrate flavored drink every 30 minutes. And also on the hour, I'd have very small amounts, 150 mils of sweet tea. It's very, very sweet tea. And every 4th hour, I would have a half of a tinned peach. Now, this was not only a treat, but it also, they slip down very easily, but the juice within the half a peach was also a glucose solution as well. So it had a calorie density to it as well. As well as that feeding strategy, we also have to develop tools, which means that the feed can get from the boat to the swimmer, because the swimmer can't get onto the boat and no one can touch the swimmer. So you end up developing these rather strange fishing reels with various implements on them in order that the swimmer can open the bottle and make that feed. So it's really important that that bottle is easy to open, even when dexterity is very limited, as it will be in a cold water swim. And also that any lines or any bottles that are used are actually fluorescent, so that swimmers can see them even at night. Okay, so let's move on to have a look at training regimes as well. So I've talked a little bit about my training regime, specifically there within the feeding, but more about the open water swimming regime in terms of, you don't just decide to swim the English Channel. Normally there's a build-up of shorter open water swims over a number of years. And it took me about 10 years of serious training to build up to this event. So again, I undertook swims in the English Channel in relays, both a two-way relay and a single relay. So you get to know the pilots on the boat and also a lot more about the swim. It's all about the research that you're doing to undertake that swim, as well as shorter swims around Jersey and also Jersey to France as well. So these are all good preparation for an English Channel swim. But other preparation include long swims, long open water swims for 8 hours, back-to-back swims at weekends. So you might do 6 or 7 hours on a Saturday and the same again on a Sunday to simulate the fatigue level that you'd be experiencing at the end of the swim. You're swimming in cold water, but also you're going to be doing a lot of swimming in a pool for interval training, as well as that long, slow distance work. Now, this training is an ideal opportunity for you to train your swimming crew to not only look at how your stroke is progressing as you fatigue, but also to train that feeding regime with your crew, so that should anything go wrong in the channel, then they are able to adapt that feeding regime and also adapt any tools to feed you as well. But also this swimming in open water is possibly an opportunity to adapt the swimmer's physiology as well. So some of the research that we've looked at around this looked again at static water immersions. And we look at this top graph here on the left-hand side, looking at oxygen consumption for a change in deep body temperature. And we know that with repeated immersion in cold water, that we can actually reduce that metabolic response to cold water immersion. And that what happens is that once we get past that deep body temperature that we've already repeatedly experienced, once that deep body temperature exceeds our body temperature that has been repeatedly experienced, we get that increase in metabolic response occurring again. So it's really important that the swimmer tries to stay as warm as possible. Looking here on the right-hand side, we have data that we compared control swimmers with cold water swimmers, both in static cold water immersion and also during a swim as well. And we see in this condition that the cold water swimmers in the static condition actually cooled more rapidly than the controls. So we see a hypothermic adaptation occurring. There was no increase in metabolism, as we suggested earlier. However, the opposite happens when we get cold swimmers to swim in comparison to a group of pool swimmers. And we see that their deep body temperature cools much more slowly than the pool swimmers' temperature. So what does this really mean? Well, it also means that it's really important that the swimmer doesn't stop too many times while swimming the channel, because our stationary cold water swimmers cool more quickly, and our cold water swimmers, when they're swimming, will cool more slowly. So That's another reason why you should keep breaks really short and also to a minimum. So now let's have a look at some of the deep body temperature data from my swim itself. I ingested a gastric pill 8 hours before the swim started, and these are the data from that gastrointestinal pill. So starting off before the swim was our transit to Dover, and then the swim is highlighted here in blue. And we returned from the French coast via a boat to Dover, and then returned to home overnight, and then had a fairly normal night sleep. Now, what really grabs me about this graph is this component here, and this rather large increase in deep body temperature, which I can't really explain. I wasn't exercising, I wasn't undertaking any particular breathing techniques, I hadn't consumed any hot food or fluid in that period of time that would have caused an interaction with the pill. So the only explanation I can that has possibly been suggested is that of anticipatory thermogenesis by Noakes, as he's noted that in another cold water swimmer, Lewis Pugh. But I'm really struggling to suggest any other mechanism than that at the moment. Okay, let's move on to have a look at some other unknowns, and that is really of our pilot. And the pilot is really very integral to the swim. They are exceptionally knowledgeable people, and they are your eyes and ears on the boat. They are the absolute unsung heroes of long distance swimming, and they're the reason that swimmers can actually make it to from England to France. We have around 12 professional pilots that take swimmers from England to France, and there is a financial cost to their expertise, but it's absolutely essential that that expertise is drawn upon. These crew, not only the pilot crew, which are the gentlemen in orange, but also your swimming crew. So these are the people that you've trained with. These people are on standby for the entire weather window of your channel attempt. And your pilot, this is the gentleman in orange here, he is solely responsible for your safety as a swimmer and also for everybody else's safety on the boat. So what he says absolutely goes. And his job is a very, very complex job. He has to be in constant contact with a number of different authorities. So that will be Coast Guard agencies in England and France. It will maybe immigration officials. It may be other boats as well. Now, the English Channel is an exceptionally busy waterway, and I liken it to the analogy of trying to walk across a busy motorway or highway. Now, what we have in this video is we have our channel boats highlighted in red, and what we'll see is a stop time video whereby we see them where we see them go, sorry about this, where we see them go all the way from England to France. And what you'll see is that all these other blobs are actually the large boats that are transiting through the Dover Strait at the same time. And they are very fast moving. They're about 300, some of them are up to 300 meters long and 50 meters wide. The ferries are slightly smaller at about 180 meters and 30 meters wide. But they're exceptionally fast moving, between about 20 knots. And so the pilot has to weave its way through this very fast moving traffic. As well as cope with this boat traffic, it also has to account for the tides and the tidal flows that are occurring as well. So here we have the tidal flows that were occurring on the day of my swim. And you can see here, this is the track of the pilot boat. And what we see here on this track on the right is the tidal flows that were occurring during the swim. So we see that the very big arrows are the fast flowing waters, and the very small arrows are very slow moving waters. And this is why a swimming track will never look straight. It will always have big curves in it, and it's because of that tidal flow. Now, swimmers are trying to aim for, and the pilots are trying to aim for this Cap Green A point here, where this red triangle is. And right at the point where the swimmer is trying to land that swim, we get some very, very strong tides trying to sweep people away from the coast and add extra time to their swim. So at this point in the swim, it's really important that the swimmer has enough in the tank to be able to sprint. And so this is why having that interval training in the tank during pool training sessions is really important. Okay, I've mentioned briefly the support crew in the last set of comments, but they are really there to support you as a swimmer. They help grease you, feed you, their moral support. They check that you are progressing well in the water, that you're okay. And they can do this by checking stroke rates. You can communicate with them and they can chat to you during the swim, and particularly during feeds. They provide great moral support. But they can also be a bit of a hindrance if they become ill, because they're on a very slow moving boat. So seasickness is sometimes an issue with being on the boat. And there have been relay attempts, particularly that have been stopped because of seasickness on the boat. Now, swimmers also have to make sure that they're able to urinate regularly, and particularly they have to urinate whilst swimming, because we don't necessarily want to stop so frequently, as I've explained previously. And that's one of the tasks of our support crew, is to check by asking, have we had a pee? Now, the reason for this is really quite important, is because if people that aren't able to urinate can be a potential mechanism for swimming-induced pulmonary edema, whereby not only have we got that profound vasoconstriction for being in cold water, but there's also a potential for overhydration because of repeated feeds, which may exacerbate any central venous pooling and cause an increase in cardiac preload. Particularly during strenuous swimming, that's problematic because it increases pulmonary arterial pressure, and therefore alveolar edema may be possible, and therefore the SIPE diagnosis. So your swim crew are equally important, and mostly volunteers who have taken possibly a week or 2 weeks off work to support your swim. So these are really key people, but they're key not only before and during the swim, but also after. These are just a couple of the pictures that they kindly took of me at the end of my swim, where I'm exceptionally sunburnt, even on a very cloudy day. And something that I hadn't anticipated was having a very ulcerated tongue after being in the water for so long. And within 24 hours of this photograph, the mucosa of my tongue and buccal cavity had basically slothed off. So not a particularly attractive look. So I had to modify what I could take for the next 7 days in order to regain taste, but also to make sure I was moisturizing the whole of my mouth. In addition to that, 48 hours after returning from the swim, I suffered some acute respiratory problems, which were recovered by oral steroids. But we really didn't get to the bottom of why that occurred, but it was my swim crew that supported me through that process. Now, not only are the swim crew really important, but there are other things that can happen whilst you're in cold water, and particularly during channel swimming. And that's really looking at the other things that could be potentially in the water. So some awareness of the flora and fauna when you go into a channel swim is a good idea. Now, I'll be polite and say it could be pollution or plastics, and also some of the wildlife as well can be a little bit curious. So in terms of the wildlife in the English Channel, it's relatively benign in terms of lots of barrel jellyfish and mauve stingers, which will give you a little bit of a sting. But if you get stung in a very sensitive place around the lips, or you may have more problems. We do have curious wildlife, so we have some seals and also seabirds that do like to dive bomb slow-moving craft because they think they're fishing boats. But also within the English Channel in particular, we have a one-way system for traffic, for boats. And so what this creates is a separation zone in the center of the channel where lots of rubbish accumulates. And within that will be islands of seaweed. And within those islands of seaweed can accumulate lots of pollution, lots of plastics, and also things that may be hazardous to the boat as well. So fishing gear may be found within that particular berg of seaweed. And if the pilot boat becomes snared with any rubbish around the props, that is the end of the swim, and the swim has to be aborted. So it might be through no fault of the swimmers that actually the swim may be aborted, and so that's why the lack of control there. In addition, there's quite a lot of paperwork that needs to be done prior to any swim, and not many people are very aware of this. So if you're going to undertake any sort of long distance open water swim, there might be some paperwork that you need to do. You might need to join a swimming association that will ratify your swim. You will need to complete qualifying swims, medicals, but also that you comply with your official observers' requests. And in addition to this, we were traveling from England to France. So everybody aboard the boat needed to have their passport, including my passport needed to be aboard the boat, as it is possible that immigration officials could turn our pilot boat round if someone didn't have their passport on them. So you see from just looking at this, diagram here that there's lots of things that we need to do before we get to the point where we can stand on the beach, either in the sunlight or in the middle of the night. So there's lots of organisation, lots of planning that needs to be done before we can then let go of all control. And the only thing we can do is swim and trust in the pilot and the crew. So I want to finish my talk by suggesting that anybody who is an aspiring swimmer, do your research. Talk to lots of people about the swims that they've done, the mistakes they've made, the things that they have learnt about. Contact pilots in particular to find out about the swims that they support. Because I am still learning about swims that could go ahead. And so there's lots of learning still to be done. So I'd really like to finish by thanking the people that helped support me to swim the English Channel. Without their support, we wouldn't have been able to make it. So that is the boat crew. So that is our Anastasia, the pilot crew, and also my swim crew as well. And in addition, I'd also like to thank not only the expertise of the swim crew, but the expertise of the scientists that have helped to produce the research that I've talked about today. So my final thank you is to you for listening as well, and I'm happy to accept any questions. Thank you very much.
Terrific, Heather. That was just a fascinating talk into not just the science, but also into, as you pointed out, the entire mass of intricacies that are involved in this. Just a clarification question from Jürgen Malau about kind of the rules or the logistics is when you're actually in the water, I guess, like, how do you communicate, and also who guides the navigation? Is it is the pilot and the support crew allowed to, basically yell directions out to you, or is it strictly up to you?
Absolutely no. The pilot has complete control over everything. They tell you the direction they want you to swim. The only thing that the swimmer can control is putting one arm in front of the other. So you've spent years, months planning this one swim, and then you have to let all control go. And so for any control freaks amongst the population, then this is quite a difficult challenge to undertake. Is you've done all the planning and preparation, and then you have to trust that that planning and preparation is going to take place how you want it.
All right, and we have another question from Gemma Milligan asking about kind of your choice of grease. Like what do you use, or what is there a standard?
Well, there's several different, most people will use a mix of Vaseline. Some people will add lanolin to that mix. Others might also add a little bit of Sudocrem as well to the mix as well. So that will give the body a white appearance. And that really is to prevent chafing from occurring. You're in quite an abrasive environment in salt water. And so you're wearing a swimming costume or swimming trunks, and any articulating joints, particularly around your shoulders, that you're going to chafe quite badly if you don't wear some form of grease.
Absolutely. And another question from Christopher Minson. I think one of the real kind of shocks, I think for many of the audience, is the kind of your body weight change and your planned weight increase and fat increase. Like what was your actual kind of experience with that? Did you have any kind of difficulty in kind of coming back down from that weight? Like how did you kind of manage the kind of your weight loss afterwards?
Yeah, it's something I'm not particularly proud of, but I can put body weight on, particularly body fat on, quite easily. I do like food, and actually it's not a healthy weight to be. So it might have been ideal for the swim, but it certainly is not a healthy weight to be on land. And I'm 11 months after the swim, and I have I've still got about 6 kilos of that body mass to lose. Clearly we're in lockdown still, and I can't get into the lab to do a DEXA scan, but I've still some considerable amount to lose. I've managed to get rid of some, but not all. So it actually takes a lot longer to get off than it has to put on.
And have you, in addition to the DEXA scans, did you take any blood measures? Did you have any kind of like biomarkers that way about kind of how your blood profiles shifted or changed over that time?
To be honest, I thought about it, and when I reflect on this, I've spoken to a number of people that say, oh, did you do this? Did you do that? And to be honest, I'm working a full-time academic job. You're training for 20 hours plus a week. You're then trying to eat a huge amount of food. You're trying to prep your crew. And so actually the logistics of trying to achieve this feat really take over from the scientific aspects of the swim. And so popping a pill is something that, yes, you can do quite quickly, but a more prolonged period of gathering biomarkers would have been great. And it's definitely something we want to do when other people swim the channel.
Yes, I think it really reflects back to kind of our talk last week with Trent and Evan talking about, you know, kind of the need for really being as least intrusive as possible to a team. And it's interesting when it's even, you know, you as both an athlete and a scientist are saying, no, I said, I'm not doing this and putting limits on that. It really brings home kind of the challenges of kind of doing this kind of research in the field with someone whose main focus, you know, isn't necessarily the science. You know, we as scientists come into this, oh, what about this? What about that? But, you know, I think it's really an important reminder to keep in mind that, you know, they have their own priorities and you can't get in the way of that. And you have to figure out how to be as minimally kind of intrusive and invasive as possible and still ideally get your science done.
Yeah, I agree entirely, and it's always one of the things that it will grate that I didn't at the time feel I had the opportunity to do that, but there are people swimming the channel every year, and so there's always the opportunity that a few people may support some additional scientific work.
All right, and I guess just to close out, I was asked, does any of the panelists have any kind of final thoughts or questions?
Yes, I have a short question, Heather, for you, if possible. And I'm interested, how much of the swimming training was done in salt water or in the sea, or how much was done in the swimming pool? Was everything in the swimming pool or not?
No, I'm very fortunate here in Portsmouth on the south coast of the UK to live within walking distance of a beach. And so the vast majority of my long distance training, the long, slow distance work was undertaken in the salt water. I trained once a day in a pool doing interval training, and then the second session would always be a sea session. So I would do roughly between 15 to 20 kilometers in a pool every week, and the remainder in seawater. So a fair proportion of that was pool work, purely to maintain speed, but also the long, slow distance stuff as well was all conducted in salt water.
Would you think it would be an issue if you were just to prepare in the pool?
There have been people that have prepared in pools. So clearly they're not necessarily very well, they're not very used to being in cold water, and that might not be so problematic for warmer swims. So the English Channel can get as warm as 18, possibly 20 degrees towards the French coast. So if someone is able to cope reasonably with the cold, but has no local facility, it's possible that pool training may be the vast majority of the work. And people have done that. They have used, the majority of their training has been pool-based. So I'm not saying there's only one way of doing it. It really depends what time you're swimming. Mine was a really very early swim and the water relatively cold. So if they have later swims in the year where the water is warmer, actually they may be, maybe pool training would be something that they could do a great deal, a greater proportion of, and still be successful.
Yeah, thank you.
All right, and with that, I think I'll close the session by again thanking both Dominique and Heather for really insightful and enlightening talks on this topic. And it's to me, it's just fascinating about how our bodies function, as all of you are interested too, but especially in this environment of cold, and very relevant to many situations. I know there's a heavy focus in a lot of places on heat stress, but cold stress does happen both for the general population and also in work and athletic settings. So again, I want to thank both of you for highlighting that for us today. And I'll just close out the session by again reminding you that if you want to go back over these talks, and also for anyone who has missed them, you can let them know that you can access them. I'll probably have them up by the end of today, up on our website, ICE 2021, and you'll see that there is a little tab there of past talks, and you can see the past talks. And again, with the links there, we also have student instructor guides to them too, and so you can access them there. So again, I will close off by thanking all of our speakers and wish you have a great rest of the week, and we look forward to seeing you back next Thursday talking about hypoxia physiology. Thank you very much and have a wonderful day.